My name is Ruben Winch, and I'm a postdoctoral researcher at the Technical University of Denmark. The project that I'm discussing today was also done in collaboration with Jeffrey Hawkes, who himself works at Uppsala University in Sweden. The presentation today centers around data reduction tools to decipher molecular fingerprints of dissolved organic matter. But what is there to decipher? Well, we have a very complex pool in dissolved organic matter. So this figure from Zach and Dietmar here shows very nicely that the material at the source consists of very few different biomolecules. So that could be lignins, cellulose, amino acids, lipids, DNA molecules. And as these molecules are subjected to a degradation cascade, the complexity increases considerably. So we're forming slowly this universal DOM component that consists of reoccurring molecules and molecular formulas that are very complex. Typically, this complexity is observed with high resolution or ultra high resolution mass spectrometry. And a data set of this kind scales in complexity with the number of samples. That could be hundreds of samples. And of course, the number of molecular formulas. We typically have about a couple of thousand of them. So we have approximately, give or take, 10 to the 5 data points. But the true complexity is even much higher because we have behind every single molecular formula the possibility of several different structural isomers. So that complexity is probably an underestimation. So in an effort to try and physically smudge apart our sample and get to these structural isomers and their abundances and their dynamics, we could employ reverse phase chromatography, as shown here in this figure from Sarah Sandron and her colleagues' paper. We have a separation into several different peaks. Here, the main peak occurs in the middle of the chromatogram, sort of a broad smudging apart of our sample. That is a response to an increasing concentration of organic solvent in our chromatography. So the polarity decreases from left to right in this particular picture. The complexity of this data set is then even higher because it scales also with the number of retention times that we're considering. So if we're doing an online measurement, we increase by about an order of magnitude. If we're fractionating, not quite as much. So what are typical and common ways to analyze such a data set? This total ion chromatogram here shows 3 different peaks, and you could choose to separate 4 different fractions. So we employ a fraction collector and say, we want the first peak, the onset of the second peak, the tail of the second peak, and then the third and last peak in the chromatogram. Then we would simply employ high resolution mass spectrometry and analyze the molecular composition of these particular fractions. But the picture is actually more complex. If you were to take the same total ion chromatogram and actually do an online measurement, you could see that you can rotate this plot around and move from the total ion chromatogram to a mass spectrum. And you get 1 for every single retention time that you can manage to measure. And if we actually also consider the molecular composition, we can see that as we are going to move in a minute here in the total ion chromatogram from left to right, from more polar to less polar molecules, the molecular composition in the van Crevelen space, so oxygen to carbon and hydrogen to carbon ratio, slowly changes continuously though. So it isn't the case that we have clear-cut fractions that differ completely between the different fractions. We have a lot of things in common and some changes in the composition, but they are very gradual. So this is a relatively complex separation that is very gradual. So a fractionation into, say, 4 fractions maybe is an oversimplification of the picture that's emerging here. So to describe a data set like this, we could combine the physical chromatography with a mathematical model that it is based on. So the typical analytical principles, we would say we have a number of analytes and the sum of the analytes makes the total chromatogram. So to consider a mathematical model, we have to first start considering the retention time. That's the inverse polarity. Then for every one of the retention times that we record, we get a mass to charge ratio spectrum that we then convert into molecular formulas. And we can do that for a lot of different samples, and then we get a 3-dimensional data set. And if we were to assume that there is a universal DOM pool that consists of molecular features that occur across different samples, we could even try and employ a mathematical model that could describe the entire data cube here. And such a model exists. It would look something like this. We have the data So that's the total observations on the left, and then a sum of different analytes describing this raw data, that it would be the model. It consists of a sum of different analytes or components. Each of these analytes has an abundance, an elution profile, a mass spectrum, and whatever this ideal model can't explain would then be left behind, or as we're fitting a model later on to the data, would be the modeling error. And if you're familiar with fluorescence, you might actually recognize this formula because it is the parallel factor analysis model. And this particular model was employed in the study that I'm presenting today. So we combined physical chromatography with mathematical separations. We were aiming to describe a data set consisting of 74 samples that was describing different headwater streams in Sweden. And a total of 2,000 molecular formulas were found in this particular data set. The mass spectrometer used was an Orbitrap, a high-resolution mass spectrometer, and we had 180 retention times. The main question of this study was, can simple statistical models such as the parallel factor analysis model actually represent the extremely high complexity of these LC-MS data sets that describe dissolved organic? And this is exactly what we tried to answer. So this was, to my knowledge, the first time that a parallel factor analysis model applied to a data set like this. We weren't quite sure what to expect and if it would work and how many components you would need, because typically we would assume that this is a very complicated data set, a very complex data set. So we might even need something like 50 components or even more. And we would maybe have to slice the chromatogram into separate fractions to describe them in separate analyses. But overall, we started by, first of all, fitting 2 components and went all the way to 10 components. And to our surprise, after 5 components, we started seeing more analytical variability. So slight changes in the way that the mass spectrometer responded to the elution, or that the elution was not completely reproducible between samples. We could start seeing these artifacts after 5 components. So we chose to go with a 5-component model, a surprisingly low number. That didn't quite fit with our expectations. And I'm going to talk about this 5-component model now. So first of all, we have the elution profiles of these 5 components. Here I ordered them according to their elution time, so not the importance to the data set, but the elution time. You can see the earliest eluting component first and the last eluting component last. And in gray, you see overlaid the acetonitrile gradient to try and make sense of the elution profiles of these components. The first one was initial response to the increase in acetonitrile, the initial increase in acetonitrile. The second component probably consisted of a delayed response to this first increase. And the third component, an even more delayed and very broad response to the change in acetonitrile in the eluent. The fourth and fifth component then probably were more related to the last increases in acetonitrile. And the fourth component even consisted of 2 peaks, one relatively broad and one more defined peak. But these were the 5 elution profiles that we could identify. Each of these 5 elution profiles came associated with its molecular properties. First of all, the mass spectrum. But what is quite clear here in the mass spectrum is that none of these components have a distinct relationship with molecular weight that is primarily seen here in these mass spectra. But they had very nice trends with regards to their molecular composition. So in the Van Crevelen space with oxygen to carbon and hydrogen to carbon ratios, you can see each component moving more and more from high oxygenation to low oxygenation, and from low H to C to high H to C, exactly what we would expect in a reverse phase chromatography like this. Now the question is, how did the model actually perform? Because 5 components was surprisingly low number. But we saw quickly that 5 components actually explained 97% of the variance that was observed in this data set. So that is, again, 5 additive non-interacting components. We can see that if we look at the bulk chromatogram and the bulk mass spectra of a particular sample, so this is just one random example taken out, we have relatively noisy raw observations, and the overall model in black seems to kind of smooth out some of this noise. and seems to approximate the bulk fairly well. So the other colored lines here represent the individual components and their contributions in the chromatogram. And we can also see at different mass-to-charge ratios, we are representing the patterns in the mass-to-charge ratio spectrum fairly well as well. So how about the things that we didn't explain well? Well, 25% of the modeling error, so that's 3% of the raw data that we couldn't explain, was actually attributable to random fluctuation. Another group that we could see relatively often in 7% of the modeling error, that was actually attributable to systematic over or underestimations. So here in the middle, you can see that the model wants to fit more abundance to this particular molecular formula in a particular sample than the raw data would suggest. So this is a systematic kind of bias. And then the last group of systematic modeling error that we could see was this case where the model was fitting an abundance to a molecular formula in a sample where we definitely did not see any abundance of a particular molecular formula. So in many cases, we actually inserted zeros to try and force the model to have no observation of this molecular formula, but it really wanted to fit a chromatogram here. So that kind of indicates that maybe we were below the limit of detection. That is the most optimistic case, but it's also possible that this is a significant modeling error. But since it was only a minor or responsible for a minor fraction of the modeling error overall, this is certainly noteworthy, but maybe not too worrisome, especially since it's the very low abundant fraction. So overall, I'd say that this model was fairly good at reducing the complexity of our data set drastically. So millions of data points were reduced to 5 components. So that makes it much, much easier to interpret a large data set like the one that we had here. It's also really interesting that such a large and complex data set with such fragile measurement techniques like online couplings of high-resolution mass spectrometry largely seem to follow the analytical principles. So things like matrix effects may not be that important or may not be as important as thought previously. But overall, we have to say that the analytical and chemical interpretation of this model is almost as tricky as the bulk chromatogram, because we saw chromatographic peaks with multiple peaks in terms of the components, or very broad peaks. So we also failed to isolate the structural isomers that we might have expected out of it. So let's conclude. We took a large data set consisting of 9 million data points and tried to employ data reduction tools according to the analytical principles at play here to try and simplify the interpretation of high-performance liquid chromatography, high-resolution mass spectrometry data sets. We were able to reduce these 9 million data points to 5 components with certain elution features and also representing different DOM fractions according to polarity. They were, of course, overlapping in their molecular properties and elution profiles, but nonetheless separate fractions. Lastly, we saw, in our case, model scores that mainly correlated with the total amount of carbon in our samples, which is nice because it indicates that this particular method actually seems to be fairly quantitative, but that this correlation also didn't really distinguish between different fractions, and at least in terms of was. But beyond this study, this kind of approach offers a lot of different opportunities, a lot of new opportunities to the study of DOM composition in aquatic environments. We can now track different DOM fractions according to their polarity, of course. That is what the HPLC separation offers. And we can track these fractions across different aquatic environments. We can identify the drivers of DOM composition. So try and find out which processes in the environment are linked to certain polarity fractions of DOM. Also, this kind of data could be used to predict the relationship between these different polarity fractions and bulk properties, such as optical properties, functional groups, and many more beyond the study